Dr. Swartz, if you want to share with us some information on ketosis and mastitis, please go for it.
All right. Thank you, Dr. Bradford, for that introduction, and thank you all for attending this seminar. As he said, I'm going to be talking about the effects of ketosis on immune function in transition dairy cows, and particularly how it relates to mastitis. So just a quick overview. As you all recall, the transition period represents 3 weeks prior to and after parturition. And during this time period, we see an increase in the incidence of disease, and this morbidity has been associated with a number of things, one of which is negative energy balance. So as cows approach calving, they start to get into an energy deficit due to drops in dry matter intake, as well as an increase in energy demand, particularly for the onset of lactation. And this becomes more and more exacerbated in the earlier stages of lactation. Also, during this time period, we see what we call metabolic inflammation, or kind of a chronic low-grade inflammatory response. And it's been shown pretty well in research that this is due to, or at least partly due to, negative energy balance. And so we suspect that ketosis plays a role in maintaining that inflammatory tone, that chronic low-grade inflammation that we see in transition dairy cows. So how do we get ketones? Well, as cows mobilize fat reserves, they liberate NEFA, or non-esterified fatty acids. The NEFAs travel to the liver, where they can go through one of 3 different mechanisms. They can either be completely oxidized into energy, which is what we would like, or they can be re-esterified into triglyceride to be stored in the liver, which is what causes fatty liver disease. Or finally, they can be incompletely oxidized into ketone bodies, such as beta-hydroxybutyrate, or BHB. We typically view these as increases in BHB or increases in liver triglyceride as an unsuccessful adaptation to lactation. So what is hyperketonemia? Hyperketonemia is the abnormal increase in circulating ketone bodies. It is defined by the measure of beta-hydroxybutyrate. We define subclinical ketosis as greater than or equal to 1.2 millimolar concentration of BHB. The incidence is variable across herds. There are some herds with quite low incidence of subclinical ketosis, but there is a nice study that came out of Dr. Jess McHart's lab at Cornell that showed 40 to 50% of postpartum cows will have at least one sample where they are subclinically ketotic. This disease has been, because it is so common, this disease has been associated with a number of other diseases, in particular displaced abomasums, but I'm going to talk more about infectious disease. So I'm going to refer more to mastitis. So starting back in the 1980s, all the way dating to present day, there have been numerous publications that have come out that have associated ketosis with the incidence of clinical mastitis as well as the severity of mastitis. I kind of like to describe the transition period as demonstrating the domino effect, where one disease leads to another disease, which may lead to another disease, and eventually leads to the cow exiting the herd. So stopping that first domino from falling, I think is key to improving transition dairy cow health. But like I said, and from the previous slide, these are associations. And because we have a saying in research that correlation does not imply causation. So if you look in the human literature, ketosis is actually quite a beneficial effect. It's been viewed that way. So the ketogenic diet is very popular. It's a high-fat, low-carbohydrate diet that has anti-inflammatory effects due to increased elevation of ketone bodies. And so when you look at the human disease models, such as Parkinson's or Alzheimer's disease, gout flares, arthritis, seizure activity, BHB reduces that inflammation, and it even improves longevity and has a reduction on anti-aging effects. So for us, this really kind of begged the question, is beta-hydroxybutyrate a causative disease mediator or an adaptive response to resolve disease? I mean, when you look at the human literature, it seems to be resolving inflammation. It seems to be a beneficial effect, but when you look on the cow side, it seems to be a causative disease mediator. It increases the risk for infectious disease. So we asked this question, and we were trying to find ways to answer this to see if BHB truly is increasing the risk for disease. So in order to understand that, we have to first realize that beta-hydroxybutyrate is much more than just a metabolite. It's more than an energy source. There's actually a few different mechanisms that it can function through. So beta-hydroxybutyrate can bind to or activate G protein coupled receptors. This one in particular would be GPR109A. It is a receptor that is on the cell membrane, and when it is activated, it has an anti-inflammatory effect. It blocks inflammation. Excuse me. There are also another mechanism called NLRP3 inflammasome. It's a intracellular sensor that exists in the cytosol of the cells. And what NLRP3 does is that it activates cytokines. It activates pro-inflammatory mediators. In particular, this one would be pro-IL-1 beta. So when NLRP3 is activated, it will cleave a peptide off of pro-IL-1 beta, which will then get secreted in its active form. So when IL-1 beta is active, it will then circulate throughout the body, and it is a known fever inducer. So it has pro-inflammatory effects. BHB inhibits this activation. It inhibits this pathway. So by doing so, it has anti-fever or anti-pro-inflammatory effects. The last mechanism would be epigenetics. So if you're really nerdy, like Dr. Bradford is, you will get all excited about epigenetics. So BHB inhibits what we call HDAC, or histone deacetylase inhibitors, or excuse me, histone deacetylase. BHB inhibits histone deacetylase, which in turn alters gene expression and alters protein function. So really what you should know from this is that it can inhibit or alter gene expression in a way to promote an anti-inflammatory environment. All right. So we tried to find different ways of answering this research question. Does BHB have an anti-inflammatory effect, and is it increasing the risk for disease, for infectious disease? So we did a cell culture experiment with macrophages. Macrophages are an innate immune cell. They are phagocyte. They kill bacteria. That is one of their main functions, is to kill bacteria. One of their other functions is to recruit immune cells into the mammary gland, such as neutrophils, to recruit other immune cells to defend against pathogens. The last thing that a macrophage does is it promotes tissue homeostasis and regeneration. So macrophages aren't just pro-inflammatory, they are both pro-inflammatory and anti-inflammatory. They have dual functions. We took mouse macrophages, it was an immortalized cell line, and we cultured them with BHB at 4 different concentrations, 0, 0.6, 1.2, and 1.8 millimolar. The 0.6 would be representative of a healthy cow, 1.2 of subclinical ketosis, 1.8 more of a hyperketonemia, maybe a clinical ketotic cow. And then we challenged them with Streptococcus uberis or not for an additional 6 hours. Strep uberis is a very common mastitis pathogen. The incidence of strep uberis mastitis is greater during dry period as well as the first couple of months post-lactation or post-calving when metabolic stress is most apparent. Okay. So here are our different treatment groups. We measured GPR109A, which is that receptor that I had talked about before that is activated by BHB. We have 6 different treatment groups. The 4 in the blue, or the blue shades, are the ones challenged with strep uberis at various concentrations of BHB. So 0.6, 1.2, 1.8, or the controls, which did not receive any BHB. Then we also have a control and an osmotic control. The control and osmotic control, neither one of them are challenged with strep uberis. The osmotic control it has a saline solution added to it. The reason why it exists is because BHB, when you add it into the cell culture media, it is added as a salt, so it has a lot of sodium in it. So in order to make sure that that sodium is not having an effect, we have an osmotic control. What I want you to get out of this slide is that strep uberis increases GPR109A. This is the receptor that can be ligated or activated by BHB to have an anti-inflammatory effect. So moving forward from that, BHB promoted an anti-inflammatory macrophage response. So we measured multiple cytokines, but the two I'd like to point out are interleukin 10, which is an anti-inflammatory cytokine, or tumor necrosis factor alpha, TNF alpha, which is a pro-inflammatory cytokine. When you look at IL-10, we had a dose-dependent increase in IL-10 from macrophages that were treated with BHB. So as you increase the BHB concentration, you increased interleukin 10, the anti-inflammatory cytokine. However, when you look over on the TNF alpha side, there was no effect of BHB on cytokine, pro-inflammatory cytokine expression, suggesting that BHB is promoting an anti-inflammatory macrophage phenotype. So as Dr. Abuelo had said earlier, he talked to you about neutrophil function. They've also done similar studies with BHB, with neutrophils. The main role of a neutrophil is to kill invading pathogens through either phagocytosis, extracellular degranulation, or neutrophil extracellular trap, as Dr. Abuelo had already mentioned. NET, or neutrophil extracellular trap, is when the neutrophil extrudes its DNA to form a trap for the bacteria. I kind of like to think of it as almost like a spider web. When a fly flies into a spider web, it gets trapped and the spider gets to eat the fly. Well, the neutrophil extrudes its DNA to trap bacteria to keep that bacteria from causing an infection. So BHB impaired neutrophil bacterial killing both through phagocytosis, which is on the figure here. It was a dose-dependent decrease, a nice stepwise reduction, all the way up to 8 millimolar concentration, as well as BHB decreased extracellular bacterial killing. What I kind of found a little bit more interesting was the net formation. So this is staining. This is a microscope image where they did an orange staining to look at DNA. So if you're measuring net formation, you're going to look for more DNA to be extruded. And you can kind of see how the DNA looks stringy. And this is the neutrophil expressing net formation. So this panel C is at 0 millimolar concentration of BHB. Panel D is at 1 millimolar concentration of BHB. And panel E is at 8 millimolar concentration of BHB. And as you can see, as you go across these panels, that net formation is being reduced, and that the orange staining that you're seeing here in E is just the DNA that is inside the intact cell, the intact neutrophil. Finally, when you get to panel F, you have a better magnification of what panel E is. And you can see the neutrophils are very much alive and intact. You can see the orange staining here in the live cells. And you can also see the E. coli. So they challenged these cells with E. coli. The E. coli was labeled with a green fluorescence protein. And so you can also tell that the neutrophils are not killing the E. coli. So I like cell culture studies. I think they help us answer research questions without much variation, but cell culture doesn't necessarily, it's risky to extrapolate from cell culture to in vivo studies where there's much more complex biology. So there's been a number of studies that I've done to try to model what we see in transition cows, try to model that negative energy balance. One of these studies was done by Perkins and colleagues in 2002, right here at Michigan State. They took peak lactation primiparous cows and they did an intramammary challenge with LPS. So LPS is lipopolysaccharide. It's a gram-negative, it is part of the gram-negative cell wall. So it mimics a gram-negative pathogen. It mimics E. coli, for instance, without actually causing an infection. It is not a live bacteria. They infused that up into the mammary gland. They had two treatment groups, a positive energy balance treatment group, which is their control, and they also had a negative energy balance group, which is the restricted group, which is in blue, where the cows were restricted for energy intake at 80% of energy requirements for 2 weeks, and then they did the LPS challenge. What was interesting that came out of this study was a delayed febrile response. So this is, they measured temperature over the course of 24 hours, And as you can see, there's a reduction right here at, I believe that is 3 and 4 hours post-challenge in body temperature in the restricted group. And then when you get to 9 and 12 hours post-challenge, there's an increase in body temperature. So there's a delayed febrile response. And if you remember when I was talking earlier about NLRP3, that is a cytokine, or excuse me, that is a mechanism to activate a cytokine that is known to induce fever. So maybe, possibly, we don't know, but that in these cows, there was an elevation of BHB, which caused a delay in that response, which delayed the febrile response. We did an infusion study just recently at Kansas State where we took late lactation cows. There were 12 of them, 6 per treatment group, and we blocked them by parity and by milk production and randomly assigned them to be infused intravenously with either BHB or saline for 72 hours. So here's an image, I hope you can see that, where the cows have 2 catheters in their neck. One is for a pump, which is right here, where we're constantly, continuously infusing a solution into these cows. We're either infusing BHB or saline into these cows for a 72-hour period. And at that time point, at the beginning of the infusion, we challenged these cows with strep uberis, another common mastitis pathogen, in both the right rear and right front quarters. The right rear quarter was a quarter that we used for biopsy. The right front quarter we used for bacterial enumeration. We also took blood samples from these cows, but we're currently trapped out of the lab, so we haven't been able to analyze that data right now. So this is a clamp. It's a BHB clamp. Again, here's the pump. Here's a close-up of the pump. Here's the cow with the catheter in her neck with the hose attached to the catheter. We are taking hourly blood samples for 72 hours. Yes, it was painful, and painful for me. We had a team of graduate students and undergraduate students who would help out tremendously, who would take these hourly blood samples to make sure that we clamped the BHB value in the BHB cows. So our target was 1.8 millimolar BHB, and we wanted to make sure that we maintained that concentration as well as we could throughout that 72-hour period. So the black line here is your infusion rate. As you can see, it fluctuates because we're adjusting the infusion to maintain 1.8 millimolar. And then the blue line represents the BHB cows, and the red line represents the control cows. The purpose of this figure is to show that we can mimic ketosis in late lactation cows by infusing BHB into them. These 2 dots right here, the blue dot and the red square, are the baseline values to demonstrate that there was no difference between the treatment groups prior to the infusion. So then we measured strep uberis colony forming units. To me, this is a really good way of measuring immune function in the mammary gland. So we infused strep uberis into that right front quarter, like I said, and then we took daily milk samples from them to enumerate how much bacteria we could recover in the milk or in the mammary from that mammary gland. And as you can see, during the infusion, there was only a minor change, but as the cow progressed through the lactation, the BHB cows got progressively more severe, and they were unable to maintain that colony forming unit at a low level, unlike the control groups that have a much lower CFU, strep uberis CFU, suggesting that the mastitis was much more severe in the BHB group than in the control group. We also measured vaginal temperature. So we have a data logger up here in the corner that was attached to a blank seeder, so a seeder without progesterone in it. It was attached to the seeder and implanted into the cow's vagina, which recorded body temperature every hour for a 7-day period. I averaged those body temperatures by day. And what you can tell is that the control cows here in red have a nice spike in body temperature, about 39.5 degrees Celsius, or which is about 103 degrees Fahrenheit, on day 2 to 3 post-calving, or excuse me, post-infusion. And then this body temperature resolves over the course of the time of the challenge. The BHB cows, however, are not having that response. Their inflammatory response is, or excuse me, their fever response is almost completely abolished during the infusion. After the infusion is over, they start to mount a febrile response. and we see an increase up to about 39.3 degrees Celsius, or about 102.5 to 103 degrees Fahrenheit, 6 days post-challenge. So as you can tell from this, BHB impairs that fever response. It slows it down, it delays it, which allows then for the strep uterus to grow in greater numbers because we have a delayed immune response. Going along with that, BHB reduced dry matter intake and milk yield during the strep uterus challenge. This was an effective treatment, not necessarily, or excuse me, not an effective treatment by time. So we saw reductions in both dry matter intake during the week post-challenge, as well as reductions in milk yield post-challenge. Kind of surprisingly, though, we did not find an effect on milk quality during a strep uterus challenge. So the somatic cell score or somatic cell count was not different between the two treatment groups. Neither was the clinical onset of disease. And by clinical onset, I mean time until we saw clots or inflammation forming in the quarter. However, there was another study done by another group, by Rupert Brookmeyer's group in the University of Bern, who did another BHB infusion study, and they did an LPS challenge. So an LPS mimicking a gram-negative pathogen challenge, an intramammary challenge. And what I want you to get out of this data is that the black triangles here are your LPS BHB cows. The open triangles here are your LPS control cows. And you can see that the black triangles are substantially less in somatic cell count than the open triangles. So the BHB reduced somatic cell count during the onset of this challenge. Now, a lot of times in the dairy industry, we'll talk about how, reducing somatic cell count is a good thing, and it is a good thing. But maybe in the context of the onset of a challenge, at the beginning of a challenge, when we need an immune response to defend the mammary gland, it's probably not a good thing. It's probably quite a bad thing. And it allows for, if this was bacteria, possibly to grow at an easier clip, to grow at a greater rate to cause a more severe infection. Going along with this group also found a greater IL-10 transcript abundance in mammary tissue. Remember when I was talking earlier about mouse macrophages, we saw an increase in IL-10 from those mouse macrophages that were treated with BHB. This data kind of corresponds with that you're seeing an anti-inflammatory response in the mammary gland, which is inhibiting or limiting host immune responses. So just some general conclusions from these studies. BHB impairs innate immune function, has an anti-inflammatory effect, particularly with IL-10. It impairs the onset of fever, which allows for the bacteria to grow, causing a much more severe infection. Particularly this was true with our strep uterus challenge. So how are we going to solve this problem? Well, there are some proven solutions to hyperketonemia. Particularly, there's prepartum monensin, or maybe you might know it as rumensin supplementation. Monensin has been consistently shown to reduce BHB concentrations in postpartum cows. And going along with that, it has reduced the incidence of infectious disease. Like I said, I like to describe the transition period as having that domino effect. So it's possible if you reduce metabolic diseases that you can reduce infectious diseases in these herds as well.